Good evening, everyone. Welcome to the spring webinar series presented by Newton Conservators. My name is Beth Wilkinson, and I will be your moderator for this webinar. Barbara Bates is our technical director. As most of you know, Newton Conservators is an all-volunteer nonprofit established in 1961. Newton Conservators is grateful for the beauty and the sustenance that the land provides for humans and the other creatures with whom we share this earth. Our goals of preserving open space and connecting people to nature require challenging the ways in which land conservation has historically excluded some peoples from protected land. Newton Conservators commits to acknowledging this exclusionary history and to building active partnerships with groups who have been hurt by that history, starting but not ending with indigenous peoples such as the Massachusetts. As naturalist Aldo Leopold wrote, "When we see land as a community to which we belong, we may begin to use it with love and respect." We're really fortunate to have Dr. Chris Richardson here to teach us about bats, diversity and activity of bats in urban ecosystems, and the status of white nose syndrome. Dr. Richardson is an adjunct assistant research professor at Boston University and a biology instructor at Tufts University. He is also a major collaborator at the Mount Auburn Cemetery Urban Ecology Research and Education Project. Welcome, Chris.
Thank you. Real pleasure to be here. I want to thank Beth and Barbara for giving me the opportunity to present to all of you a little bit about, first of all, what are bats and why they're important, and also kind of the research I do and how that hopefully gives more insight into what's happening to bats and their role in our world, in our ecosystem. So, as Beth mentioned, I have a position at BU. I'm also connected with the Broad Institute, and I also teach part-time at Tufts University. So my title is Ecoimmunology and the White Nose Syndrome. And I changed it kind of again at the last minute because I kind of think in the end I want to emphasize more today the broader question of what is happening to bats as far as white nose syndrome and how that ties into a very exciting discipline that brings together ecology with immunology. And I definitely, hopefully, will have time to also talk about the urban ecology I do on bats. And I think there will be time, but my main emphasis is going to be more on this side of my research and my background. Okay. So I thought, first of all, I put up a nice picture here of a very common and important fruit-eating bat found in Australia. It is part of what is known as a megachiroptera. So they're kind of two, so bats are part of the order Chiroptera, which means hand wing, because their wing has evolved from essentially the equivalent of what we might think of as our hand, right? The digits of their fingers are in many ways much more elongated than the typical finger of a human, and the wing has evolved to have membranes that have come out in between each of those digits. And so if you think about bats, they're hanging on their thumbs basically when they decide to hang upright, as opposed to hanging them upside down when they hang on their back feet. So there's the Megachiroptera and the Microchiroptera. The Megachiroptera are like this fruit bat, or this fun picture here I got recently from a friend. Here's a fruit bat, same species, carrying a pup, and they can do that actually. And to some distances, they can carry their pups. And Megachiroptera use more vision to look out for fruit. They typically are more fruit eaters. They have very limited to no echolocation. I'll say more about that later on. Whereas Microchiroptera, oh, let me show a picture here. I'll just jump ahead of the picture. Oh, well, okay, I got that screwed up. I'll show you Microchiroptera later on. But Microchiroptera are typical bats, say, in the northeast of North America. So those might be your common big brown bat, a little brown bat, which I study. They are pretty much all able to use echolocation, with some exceptions. And echolocation is the ability to generate high frequency sounds, so a much higher frequency than what the typical human ear can hear, to navigate their environment. And they don't require vision to use those sounds, right? Those sounds help them determine how close objects are and how fast they approach when they're flying. Um, Microcoroptera are found pretty much on, I think, every continent except for Antarctica, whereas Megacoroptera are principally found in the South Asia, Africa, and parts of South America. Um, Microcoroptera are much more diverse in what they eat. The majority of them are insect eaters, although some eat frogs, some eat birds. There's an amazing type of bat that lives in Europe called a noctule, and that can actually grab birds right out of flight, songbirds, and eat them. So it's pretty wild. Although birds will definitely prey on bats too. Um, some of them also, of course, drink blood. The infamous vampire bats are part of the Microcoroptera, and even some eat small frogs or reptiles. But the majority eat insects, and they eat insects they catch in flight. Okay, so like I said, they use high frequency sound pulses, the Microcoroptera, to navigate their world. All right, now what do bats do? Right, it's worth taking a moment to think about that. Well, bats are very important ecologically in several ways. Number one, many of them pollinate flowers. Particularly true of the Metacoroptera, but they're definitely Microcoroptera as well. Um, Disperse fruit, dispersing the seeds after you eat the fruits are very important seed dispersers, especially in the tropics. Ecologically, bats, particularly the Microcoroptera, are very important in consuming insect pests. There's been studies that have indicated that maybe 300 to $400 million a year is saved by not using pesticides as far as just the role of the typical Southwest bats, like the Mexican free-tailed bat. They eat a large amount, and there's many of these bats eating every night cotton boll weevils, another pest. And that, of course, reduced the impact on crop production. A recent study also was done just several years ago in, I think it was Indiana, that demonstrated that the big brown bats definitely eat important insect pests, including invasive insect pests like beetles and certain moths, and that their presence reduces the number of those that otherwise would damage trees. So they have importance ecologically and economically. Um, so then I thought it'd be worth bringing up, well, why study bats? So, particularly, why do I study bats? Well, from the perspective of what I would describe as ecological physiology, right? That's the study of questions of physiology in organisms like birds or mammals in an ecological context, right? Bats are really just ideal models. They're excellent models for examining and thinking about the kind of factors that influence how energy use varies in particularly vertebrates, including mammals and humans. They're really useful for understanding overall physiological function. Um, so many people now study bats for their way that they can lower their body temperature, the way they can lower the metabolic rate. There's a lot of fascinating changes that bats experience in terms of hormone use that we're now studying to understand more about our own endocrine system, our own hormone system. Um, Bats are facultative endotherms, right? That's a big word, but endo means internal, right? From inside, endotherm is heat source. And that's because, like all endotherms, basically all birds and mammals are endotherms, um, endotherms rely and have access to a kind of constant, steady internal heat source. And that heat source comes from the minimal metabolic rate that is necessary to keep the organism alive, to repair its tissues and keep its organs functioning. And with the availability of insulation, like feathers or fur, that allows for the ability to maintain a constant body temperature, at least for much of a given part of the lifetime of the organism. Facultative means they can adjust their metabolic rate or body temperature in a way that we cannot do. Humans, um, are more obligate endotherms, right? We can't lower our metabolic rate or lower our body temperature. So that means bats can do quite a lot through how they physiologically affect regulation of temperature. And that means both in daily situations and seasonally, the typical big brown bat, Aptescus fuscus, a little brown bat, Myotis leucificus, or little brown myotis, depends how you want to call them. They can hibernate, of course, part of the year. And when they go into hibernation, they experience a number of really interesting changes. And there's a lot we've learned about those changes that give us insight into how hormones are used in energy use, hormones are used in controlling metabolism. Um, Also, we've learned more about how, as bats come out of hibernation and go into reproductive activity, how that changes physiologically, have given us insight into just how reproduction works. Um, Also, immune function. When bats experience torpor, they typically do what's called down-regulate immune response. They turn down their immune system. It doesn't activate as easily. The reasons why they do that, and then they have to turn it back up. Studying that phenomena has given us insight into how certain humans, for example, react dramatically to certain kinds of diseases when they're already immunocompromised, right? And they experience another disease. And overall, just how bats thermoregulate has given us a lot more insight into thermal biology in mammals in general. All right, so the point is, bats are very useful to study in terms of understanding not only, again, metabolic rate and physiology, but also how they respond to diseases. So, one of the big things that, of course, occurred during COVID is more people got to learn about how bats can carry viruses. And of course, since that time, more people are paying attention to bats and viruses. Now, we still don't understand exactly why is it that bats in many parts of the world have a high percentage of viruses, like much higher than a typical mammal. Now, some believe that's because there are high temperatures they can generate when they're in active flight, allow them to tolerate and check virus activity. Others believe the fact they can drop into torpor, at least many of them, allows them to keep viruses in check. It's still not clear. For example, in North America, we know there's a number of coronaviruses that bats carry. Fortunately, we don't have any reasons to believe they can harm humans. But part of my research, in fact, I remember in 2019, right before COVID and into 2020, I was talking to a virologist over at the Tufts Veterinary School about my interest in looking at the connection with the flu, because he studied the flu in marine mammals and how it spreads. And he was interested in my proposal that maybe viruses could impact how bats are dealing with white nose syndrome, which I'll say more about soon. Anyway, I was already interested in this question of viruses in bats, and all of a sudden COVID hit. The next thing you know, people are talking to me about my developing research in viruses in bats, where we're looking at what viruses are present in bats in the Northeast. Okay, so to dive a little more into the science, in the science I do, and why it has some important implications for ecology and conservation. Let's first define a little bit more what is ecological immunology. Well, the name tells you something, right? It's a discipline of immunology put in the context of ecology. It really is, in some ways, trying to understand how immune function and immune response is impacted or impacts the environment animals are in. So there are several important focuses of ecological immunology. One is, what is the source of variation? Why do different birds, for example, where there's been a lot of research on the ecological immunology of birds, why is it that variation among birds, say, and now we're learning more about with bats, is connected with differences in stress, differences in diet, right? So a key part of ecological immunology is understanding how variation exists and why it exists um, among individuals in immune function or among populations. And that brings in topics of environment, right? Life history trade-offs. It's well known that animals in the wild, like whether it's birds or definitely bats, if they invest more, say in immune response, that might reduce their ability to effectively reproduce. In fact, part of my research has been better understanding when and how that happens. For example, we know that birds are more vulnerable when they're molting, when they're actively generating new feathers to parasites, and that can impact their reproductive capacity and how many eggs they can lay. At least this is birds in the temperate region, which I'm a little more familiar with. Cost immunity, right? Cost energy to mount an immune response. It costs energy, it costs nutrients. Like you have to spend, the body has to put energy to put together proteins, right? There's issues of repair, right? It's costly. So knowing how much these things cost as far as immunological function and how the environment affects it, another part of this discipline. The role of environment thus brings me into urbanization. Like I said, I'll have time more to say later, a little more later about this, but basically we're learning more that, for example, in areas that become increasingly urbanized, immune response definitely changes. This has been demonstrated in birds. Preliminary data suggests this is true in bats, but how it changes is complex. We're still trying to understand it, but definitely the environment can impact immune response. Um, in fact, stress itself is known to be something that can reduce immune response. Chronic stress is known to make, say, I was reading the other day about elk in the western states who are more vulnerable to a type of virus. And this is basically a wildlife disease when they're put into more dense, stressful conditions. And then finally, trade-offs. Like there's a very complex interrelationship between different parts of the immune response, right? We have our classic antibody response that can be induced by a vaccine. You have your inflammation response, which can happen almost anytime you experience a pathogen challenge. And the energy and the nutrients that are put into each of those components can change in the life cycle of the organism. And that includes bats. All right, so I got more into the topic of bats from the perspective of, well, what is happening with bats and white nose, and how can we learn more about it to help bats survive and recover. And so that's kind of how I got into ecological immunology, through helping better understand white nose syndrome in bats, because we're learning more than every day about how they respond to the disease. And in fact, the broader disease ecology, where you bring more in the pathogen along with the bat, the host of the pathogen, has become more of my research recently. Disease ecology of bats and the fungus that causes white nose. All right. I wanted to say a little bit about energy expenditure. I'm pitching this at a little more of a scientific level, but I've been told that many of you would appreciate that. So all organisms have to spend energy. Actually, I'm going to just hide my controls. One second. Okay. So it's known, in fact, that energy expenditure rates, like how much energy you spend every day to stay alive, or how much energy you spend when you start to run, or how much energy you spend when you're dealing with an infection through a fever, right? Which can vary among individuals. We're learning more about how decent, cutting-edge medicine should recognize that not everyone's the same. There's variation between biological men and biological women. There's variation amongst people by age. There's variations amongst different individuals and how they respond to diseases and how they should be treated. And a lot of that can be due to differences in energy expenditure. Now, one of the kind of hallmark features of energy use and metabolism is that all mammals must put in energy to properly maintain their tissues and organ function, to properly maintain overall balance, which is known as homeostasis, and not to go into pathology or disease. They have to spend energy on that first and thermoregulation, maintaining a proper body temperature, before energy can be made available for any other function, including reproduction or growth. Okay, oh, here's now the picture I was going to show um, of the little brown myotis. This is one of my main species I study. This is a fairly common bat found throughout much of North America, although definitely since white nose syndrome, their numbers have been dramatically reduced. The good news is, I'll say more about this, they are recovering, although unevenly. This is a local hibernacula in central Massachusetts, where I recently was studying little brown myotis, try to learn more about how they use hibernacula, what like temperature is the ideal temperature for them. Does that temperature vary? I don't know if my cursor can be seen, but um, basically we believe that even between this spot, the bat in the top right and the bat in the lower left, there can be variation even in small ways in temperature, and they might pick different local temperatures and microtemperatures. So in other words, I'm playing a role in learning more about how important microclimate is to optimal hibernation conditions. Here's one of my favorite pictures. This actually goes back to my PhD days. This is in Palmer, Massachusetts, out near Amherst. These are big brown bats. I'm looking up at them. They're roosting up in a small barn. This is taken, I think, in May. Typically in May, coming back by April, little brown myotis and big brown bats become more common in New England, typically taking up residence in either attics or barns. The females cluster and they're called maternity colonies. This is a maternity colony. Here is some big brown bats. See them up in the top center, roosting in a bunker, actually. There's a number of these old World War II bunkers that are scattered all up and down the coast of New England, all the way down Cape Cod, all the way up to Maine, and they're pretty much abandoned now. And in some places, they become vital spaces as hibernacula, where bats will, if left undisturbed, will basically live during the winter during hibernation, like the big brown bats here. Okay, so this is a tricolored bat. It's a pretty unique bat in Massachusetts. It's definitely threatened. You'll notice that it's called tricolor because it has this kind of light pink along its forearm, contrasted by some kind of orange and off-color brown, and a little bit of a kind of a yellowish on the underbelly. Thus the name tricolor, three different colors. These were found in a hibernacula that I recently visited this past winter as part of the ongoing study on what is happening to bats during the hibernation period. And one thing to know about the tricolors is that they're doing okay, but they could be doing better in terms of how they're surviving and recovering from white nose syndrome in New England. And this is me in a hibernacula in Massachusetts again. It's very close to the hibernacula I just showed you in the previous picture. I'm actually holding a little brown myotis, a little brown bat, sometimes called. You'll notice I'm kind of wearing the full Tyvek gear. That's mostly to protect or reduce the chances that I could be bringing PD fungus to the bat. There's no real diseases that bats in the Northeast can easily give to humans. It's more of a concern about bringing that fungus from one place to another. Thus, the Tyvek protective or kind of covering. Little brown myotis also hibernate in the same hibernacula as the tricolor bat. Thus, we were in the same area this past winter again to understand more about how little brown bats are using hibernacula and how they're responding to white nose fungus during the hibernation period. Now, just say a little bit about white nose. So, white nose syndrome is a disease that is caused, we now know it's caused by a cold-loving fungus, which basically means it has to have somewhat cool temperatures to survive. I'm not going to tend to try to be able to say the full Latin name. We just often go by PD for short. And around 2006 and definitely by 2007, we started to realize this fungus had taken hold first in eastern New York and then soon throughout New England. And it's led to the death of probably at least a few million bats. The good news is bats are recovering, although it's an uneven process and some are very vulnerable. White nose has affected some bats more than others in North America. Little brown myotis is starting to recover from white nose syndrome. And the big brown bat, generally speaking, the one I mentioned earlier, has been doing pretty well overall in how it's responded to white nose syndrome. But basically, the idea is we capture the bat, we take a little like almost like a Q-tip, and we kind of gently swab up and down the forearm with little bit of water on that Q-tip, distilled water, and then the idea is we're trying to pick up any of the fungus. And then later on, we do what's called qPCR, quantitative PCR. It's a way to detect and amplify particular types of DNA. In this case, we're looking for the PD DNA, and the idea is the more fungus present on the bat, the more of that DNA we pick up, and that becomes a way to assess what's called fungal load. Um, in general, we've learned that, bats that have the fungus will tend to arouse from torpor more often. It tends to disrupt because it disrupts their torpor. It then tends to make it more difficult for them to survive the winter because they end up using valuable fat reserves. Now, characteristics of white nose syndrome are, of course, first and foremost, that's the name, the white fungus. It's found in the nose, the ears. It can be scattered around the wing membranes. We also know now that the fungus can definitely be present even if you don't see white. That's why it's very important that we actually swab the bat to assess for level of the fungus, because sometimes it's not obvious how serious the white nose syndrome is for that bat at that moment. Another feature of white nose syndrome, as I mentioned a little earlier, is that over time it causes the bats to use up their white and brown fat reserves. Thus, they're at risk of essentially starving because they can't make it through the winter. Um, they also have reduced ability to go into deeper torpor because as they use up their fat reserves, it becomes more difficult for them to come out of deep torpor. In general, their ability to respond to hibernation, I mean, to respond to the immune challenge is reduced, thus their immune responses are disrupted. One other thing we've learned over time is that wing damage can be quite serious, but we've come to realize that it creates what's called evaporative water loss. So the more the wing is damaged, the more they lose that precious membrane, the more there's increased risk of water loss. Or if the membrane is still present, but it's disrupted by lesions and other kind of chronic damage due to the fungus's attack on the wing, that can also lead to increased water loss. Scarring damaged wings are a hallmark, and it's now believed that bats in some cases can die from dehydration before they die of fat deprivation if they experience white nose in too much of an intense way. All right, I'm going to show you some pictures taken in the beginning by my mentor, Thomas Coons, and some of his colleagues, some of his students and my colleagues, John Riker, Miriam Moore, and Nate Fuller. I did my PhD, my postdoc at Boston University, and that's how I got into bats and understanding bats and white nose. So what you see here is cluster of bats, a good sized cluster, that's at a spot typical at the top of Aeolus Cave. Aeolus Cave is one of the most important hibernacula in the whole Northeast. It's found in southern Vermont, up in a mountain, and it's estimated that at the peak, before white nose, there could have easily been 2.5 to, sorry, I was going to say a magnitude of 10. I meant to say 250,000 to 500,000 bats. Right now, they're pretty comfortable and they're down to around 100,000. So still quite a significant number of bats that hibernate there. But what you notice is that whereas in previous years, this also be covered with bats, within 1 year of white nose, oh, I'm sorry, this is just how many bats were present there, how many weren't present just from those that were dying. There is a picture of a bat taken right out of ALS. You can see the telltale fungus on the nose and on some of the ears and on the wing. And there is what they found. Tragically, many of the bats is starved to death and then froze to death, would never get out of torpor. It was definitely a devastating time for all of us, those who really studied bats. Now, the good news is bats are recovering, but it's uneven. So, just a few things more broadly about bats now in North America and how they're responding to white nose. It's been confirmed that 12 species definitely have white nose. Six other species have the fungus, but no obvious presence of the disease. Although I think in the past 2 years, six of those species now do have two of those six now have the disease. The bats that have been most impacted, at least originally, by white nose in a very negative way, are the tricolor bat, the little brown bat or little brown myotis, um, the long-legged bat, and the northern long-eared bat. Um, the bat that's probably been most successful at evading the worst impacts of white nose is the big brown bat. In fact, in all the years of studying them now, I've rarely caught big brown bats with serious damaged wings or seriously unhealthy bodies. So there's several scientific hypotheses about why, and I guess even say theories, to why big brown bats are doing very well. One possibility is they tend to have the smallest cluster sizes when they hibernate. So it's possible they're not spreading the fungus as easily amongst each other. Physiologically, they tend to be more robust in terms of energy use. They tend to have, at least when adjusted for body mass, a lower metabolic rate. So they might be more effective at conserving energy. That's another developing idea, and I played a role in that research. In general, the belief is that bats in New England and the Northeast in general, and now the rest of the country, as they're picking up white nose, are vulnerable because, first of all, where they live is an optimal climate for them. The microclimate is an ideal set of temperatures and humidity for bats, is also ideal for the fungus. Also, bats are, again, very, limited on the energy resources. They're experiencing this fungus first and foremost during hibernation when they have to rely only on their fat reserves. Now, I will note right now, and I'll say more about this later on, but bats basically deal with the fungus and white nose syndrome in two major parts of their life cycle. First and foremost, during hibernation. That's when they are at risk of death. That's when mortality is connected with white nose. But they also are impacted during reproduction when they're coming back after hibernation. And they can still bring white nose with them or pick it up from each other, at least the fungus. But rarely do they die during that period because they have active energy. Sorry, they have active real availability to energy sources. They have insects they can feed on during the spring when they're dealing with white nose. So the impact is more on reproduction and less on their survival. But I'll say more about that when I talk a bit more about some of my research on reproduction and immune response of bats and white nose. But in general, the way they cluster, the way they swarm, it's very easy for them to transmit the disease. Now, what does it mean to say naive? Well, what we've learned, there are two main type of PD strains in Eurasia. And it's believed they've been there for thousands of years. And one of them was brought over, in fact, by cavers. We've now recently confirmed this. There's been a really interesting study that showed the link between the genetic makeup of a fungus found in the New York caves where it first spread um, to areas in Ukraine and I think Poland. And it's not, say the fault of these cavers in the sense that at the time they didn't know anything better. And they were not the type of cavers who were unrespectful for the cave habitat, but they just brought it over across the Atlantic, we now believe, and that's how they got it. Um, PD2 is not over here yet, and everyone's hoping to keep PD strain 2 out of North America and keep it in Eurasia. So there's a lot of protocols now about protecting caves. Point is, PD1 came over and no bad experience it before, and thus no immune response, no immune history, and thus they're very vulnerable to it sweeping through the population. Now, this chart's trying to show you something very interesting. If you look at the lower left, you'll notice how in the typical normal pattern over days during hibernation, there's, arousals maybe every 10, 20 days. Otherwise, they stay in torpor pretty consistently. But during white nose and post-white nose, research has shown they tend to arouse more often, especially near the latter half of hibernation when they're most vulnerable. You can see right here, from days like 50 to 80, there's maybe 2 or 3 times more arousals um, in the post-white nose period than there was in the normal uninfected hibernacula. Basically, again, increased arousal means more likely to burn up their fat. When you arouse for hibernation, typical mammal muscles don't function. They're just too cold. They rely on something called brown fat, which is found around their upper back, around their neck and heart. And that fat is pretty remarkable. It has the ability to immediately start to heat up by oxidizing the fat and just generating heat. And that can quickly warm up the fat, warm up the muscles, and then they can begin to shiver and go up to complete normal temperatures. As I mentioned to you, we're learning that with more wing damage, they have more likelihood of water loss. They're more able to experience dehydration. And of course, just overall, white nose has led to behavioral changes that's also made bats more likely to be exposed to white nose. Kind of a positive feedback loop in a bad way. All right. Now, the purpose of this is to highlight that there's been a lot of collaborators. I mean, look at these authors. All these different authors are various people. I know some of them. They're biologists, state biologists, um, federal biologists. This is one of the first big papers done. I think this is around 2020 to really assess the extent at that point of where white nose has gone. For example, you can see red is the confirmed, see the big brown is the gray. So they cover almost all of the US and parts of Mexico and parts of Canada. Red is where, white nose is gone. If you look at the little brown, much of the gray up in Canada is now areas with white nose. So I'm sorry, that's the northern I meant. With the little brown myotis, there are areas of the range where they don't have white nose yet, but a big assessment was done, very useful assessment about the degree of spread of how far white nose had gone, with extent of situation. It was a really important study that involved U.S. Fish and Wildlife Service. It was called the white nose syndrome. You see in the bottom lower right symbol. It's called the white nose syndrome response team. Um, John Reichard, right there, and Jeremy Coleman are two very important individuals who work the head of this team. They're part of U.S. Fish and Wildlife Service, um, and they're very, very important in pulling together a national response and they're still playing this role today in understanding how to deal with white nose. By the way, Tina Chang is the current leader of the Bat Conservation International in terms of the role in understanding white nose from the role of BCI and contributing to that as well. All right, so what's the impact again of white nose on a bat? That was a big question I was starting to come up with. In fact, I started wondering around 2017, 2018, how do bats individually, how their response to recovery affect their energy use, affect their metabolism, affect their long-term and short-term immune response? And what might that mean to their survival and reproduction? And I started asking questions, could alterations or changes in energy use or metabolism and overall immune function affect future response of bats to the fungal pathogen, right? So just briefly, it's worth stepping back and giving you a little review about what is immunity? What is the immune system in a mammal? Well, first of all, there's two major systems or two divisions. There's the innate, known as the also non-specific immune response, and there's the adaptive or specific immune response. And innate basically is as it describes. It's all the immune responses that are part of um, dealing with any pathogen. Doesn't matter if it's a bacterial, fungal, viral. It's the immune response that mammals mobilize whenever they're under attack from a pathogen. So inflammation response, fever. That's right. Fever, while painful, is a very important response to stop a pathogen. Those are all generalized responses that are part of the innate system. Complement protein, I'll say more about that. Complement protein is a very important way that mammals use a protein in their blood to respond to pathogens in general. There's the adaptive immune response. That's the T cells, the B cells that we all know about. They're an important part of also responding to specific infections where there's a specific B or T cell or specific antibody to a particular pathogen. And that can be used in the future as well after exposure. Thus, the idea of vaccines, right? Vaccines induce an immune response that then leads to immune memory that allows that host in the future, if they come across the pathogen again, to rapidly respond to it. But basically, innate immunity is a very important type of what we call early response, early line of defense against a pathogen. And that's what I spent most of my time studying in bats. And one very important response is what's called complement proteins, as I just mentioned. Complement is really fascinating. It's probably evolved, we believe, over 400 million years, even before mammals were on the rise in early mammals during the age of dinosaurs. And what these proteins do is they set off cascades where one activates the other, and that leads to various things they can do. Among other kind of cool things, they can put holes into pathogens that get into your blood. They can help what are called macrophages that roam your body looking for parasites and pathogens to engulf. They can help the macrophages find that pathogen. Like my favorite macrophage, sorry, my favorite what's called phagocytic cell, that which can engulf is a macrophage. I'm sorry. That's my favorite type of immune cell. And there's other things that complement proteins can do that are important in the immune response. Now, just again, I mentioned before, but I don't want to leave anything to chance. If you have, and it's known, and it's definitely known that there's energy costs and nutrient costs to immune response, then if energy becomes limited, how energy is allocated to immune response can be impacted if energy has to be allocated somewhere else. So it's been studied recently, or at least I know recent research has confirmed that human women, biological women, when they're pregnant, as they go through gestation, sometimes may put less energy in the developing embryo or fetus um, if they're experiencing infection. Now, how serious that consequence is still not clear. That's been confirmed again recently in humans. How often this happens and in what way it happens is controversial. It's believed that some birds and mammals will spend more energy on immune response and give up reproductive success to survive if it's really life-threatening. Some will risk more serious impact of illness in the hopes to have successful reproduction. So they're basically trade-offs that are known to go on. And the question is, how's this playing out in bats that are dealing with white nose and then during reproduction after hibernation? All right, so that brings us to after hibernation. Now, I want to tell you a brief little story very quickly. His name was mentioned, Nate Fuller. So unfortunately, my mentor, Tom Coons, got struck by a car and was unfortunately required to retire from research in 2010. And at that point, we knew that white nose is still very serious. So Nate at that time was trying to finish up his PhD, and I was finishing my postdoc. And the chairperson of the biology department agreed to kind of help Nate and these two other graduate students and me kind of finish up our projects. At that point, Nate and I started collaborating and talking about, everyone's talking about white nose during hibernation. And we knew that Tom knew that it was an important question to also think about after hibernation. And he was hoping to focus more on that. So Nate and I decided to spend a little more time on what's happening to bats after hibernation as far as white nose. We know now that as they emerge, many of them are still dealing with the fungus. Either brought it with them from the hibernacula or pick it up in the maternity colony from other bats that brought it over. And they may have already defeated white nose, and here they are dealing with it again. And that could mean up to 2 months that they're spending energy recovering from the disease. Typically, it's like, May into June. That costs energy. Meanwhile, all these female bats are coming back. Big brown bats, little brown bats, northern bats, tricolor bats, and they're going to pregnancy. And that costs energy. And all these bats eat insects. And they come back like right now, on a cool day. Insects are around, but not as active on a cool night, right? Insect populations typically aren't very high in early spring. Their fat reserves are limited. In other words, I gotta tell you, it blows my mind. These bats are like superheroes. I mean, they come back, these females, they have limited fat reserves, they have a developing embryo, they may be fighting a disease, they're trying to go out and fly, get energy. It's amazing how they survive, and they do survive. And I'll say more about this. Um, so you can see there's some serious energy challenges these guys face when they come back after hibernation. All right, just briefly, I'm not going to get into all of it, but I thought it'd be fun to show you just some real hypotheses I've been developing. I presented some of my findings on these hypotheses. I'm working on a paper to publish on this. I've given public talks on it. But basically, one of the key questions was, is it true that bats that experience higher PD fungal activity are experiencing higher energy costs? Will that break down as they go further into pregnancy? As a bat spends more energy on pregnancy, they may not be able to spend more energy on fighting the fungus. Right? That was an important hypothesis. And we have suggestion the answer is yes, which is important to know. That in fact they are risking more serious impact to the fungus by spending less energy on the fungus and spending more energy on pregnancy. Another big question was, and a prediction hypothesis is that with bats that are recovering more slowly and are experiencing a more negative impact from white nose on the colony level, I predicted that metabolic rate would tend to be higher. And bats that are growing more effectively, their minimal energy cost, their basal metabolic rate would be lower. They're more energy efficient. And that means they have more energy potentially to devote to developing embryo, to lactation when the pups are born. Thus the ability to more robustly respond to disease at the colony level. That's hypothesis 2A. I won't say much about 2B, it's complicated, but there was questions about immune response as well. And by the way, BKA means bacterial killing assay. I realize I didn't introduce that in the slide. I'll say a little bit more about that later on. But that's the way we look at how well common protein works. And I'll say more about that in a moment. But basically, you expose bat blood to in this case, of bacteria. It's kind of cool. And see how well the bacteria grows. I'm working on a version of it where we expose bat blood to the PD fungus and see how well different bats' blood or plasma taken from the blood it can stop the fungus. Now I want to show you the scale of impact from the disease. This is basically a very severe damaged wing. My system of measurement with my colleague Catherine Innocent that we published was scored this as a wing score 3. This is hallmarked. Beth, real quick, can you see my cursor?
Yes, we can see
Okay,
your cursor.
great, thank you. So you can see right here, these are lesions. There's lesions, those are lesions. Wing score 3 is the most severe state, one of the most serious level of impact. The pathogens causing all these lesions. There's also a lot of what's called black necrotic dots, all these dots, active necrosis, tissue damage. The fungus we know now, is actively penetrated beneath the epidermis. It causes what's called cupping erosions, hyphae, which are little extensions of the fungus, are in there. The bats are mobilizing in response to it, and part of the response, if inflammation, is leading to damage. Also, notice how there's what's called wing recession, and you can see how the wing is tensing up here. Oh, this is called wing score 2. Still a lot of black dots, a few lesions, not quite as severe, a little bit of wing recession. Typically, we don't see wing score 3 bats in post hibernation. That's maybe more during hibernation. We see these though, like right now. If I was to go out now, which is a little bit early in the season, you'll see more of these bats. Also, you'll start to see little tiny white halos kind of forming on the black dots. And also you'll see holes too. Those halos are the really beginnings of healing. Wing score 2 severity, or we call severe damage still. an active response to the fungus. Then we have wing score 1. Notice all the white spots and white areas. These are areas of depigmentation where fresh new tissue is coming down and it hasn't quite brought back the normal pigment. This is during the phase of healing. You still see some black dots, but more recovery here is going on and more energy is being spent on recovery and repair. And then there's a healed wing. Notice, by the way, tiny little scars are still left over. This is definitely a bat. We know this for a fact. We caught this bat that actually had a serious wing damage when we first caught it. That's pretty much healed. And they can heal. It's pretty amazing. Wing score 0. All right, so there's a quick summary of it. The two main areas, wing score 2, which is called subnecrotic, large number of black necrotic damage, little initial healing, white lesions. Fungus is being actively pushed out. They're actively fighting the fungus. Active resistance. Wing score 1, more recovery, more white spots. Now, briefly, how do you measure metabolic rate in a bat? And what is it? So, I once had the unique pleasure of being over in Beth Israel. They have a pretty impressive sleep center. And part of their sleep studies for those who are staying overnight is their controlled temperature and oxygen concentration rooms where they have complete control over oxygen concentration and carbon dioxide levels, and they can measure metabolic rate. They can measure how fast the humans using oxygen consumption or using oxygen, sorry. And so one way you can measure metabolic rate is you look at the mammal, they're resting, they're calm, they're what's called post-absorptive after digestion is done, and they're not cold stressed or heat stressed. And that becomes a very good standardized measure of what is the minimal metabolic rate required to keep that organism alive, right? And I measure that in bats. Now, maybe you'll see something here that's fun about this picture. I've kept this picture now for almost 10 years. I discovered when I made it that maybe you'll notice the bat's wings are sticking out on both sides. Of course, that's not how it works. This is a contained chamber. It's a sealed system. Basically, air is being pushed through the bat. They breathe oxygen routinely, and this oxygen analyzer can pick up on how fast they're removing oxygen from the air. I know how fast air is being delivered to them based on my pump, and I can measure the flow rate and then the difference in oxygen concentration on the inlet versus the outlet, and then we can estimate oxygen concentration as an indirect measure of metabolic rate. This is called respirometry, measure of respiration. And it's an important way you can at least indirectly measure aerobic metabolism, which is an important indicator of basal metabolic rate. Immune testing. The main way I did it, as I already mentioned to you, is I took bat blood or bat plasma and I exposed E. coli to bacteria and saw how well the E. coli did when exposed to um, the bat plasma. And as you may or may not be surprised to know, bat blood is pretty impressive at killing bacteria. They have pretty powerful antimicrobial activity because they come with protein. So this immune measure I did, the bacterial killing assay, is one important way to assess how effective bats are at mounting the complement immune response. I also measured white blood cell by looking at stained whole blood, and we do white blood cell counts as another measure of innate immune response or immune function. Just thought I'd throw up the statistics. You know, some of you may have heard of stats, and some of you maybe are more familiar with it or less familiar with it. But I did a number of statistical analyses, and like any kind of statistical analysis, you're looking at evidence of an effect, and that just suggests that questions about are wing score 1 versus wing score 2 different? Is there statistical evidence that suggests that's a real difference? Are there differences amongst the colonies? Is there differences in fungal load relationship to BMR that matter? Now, my results were kind of cool. What we found is that after accounting for the effects of body mass, bats in the second half of pregnancy during wing score 1 in colonies that were at a lower growth rate tend to spend more energy. as I predicted. And that's what the Ernst colony is. It's a much lower growth rate of colony growth than the Sing colony. And that was very powerful evidence that, in fact, more energy is being spent by bats that are not doing as well. Also found out that wind score one bats tend to have higher energy expenditure than wind score two, which is really kind of exciting in the early phases of pregnancy. This is just a kind of cool picture. The red dots or red triangles represent the low growth colony bats, the Ernst colony. The black circles represent the high growth. And you can see very clearly that when you account for the effective biomass, like look at the lines, the one line is basically higher at all points than the other. In other words, the Ernst bats, the low growth colony bats, tend to spend more energy on average, in at least as measured by oxygen consumption rate, than the high growth bats. All right, so let's take a message. Basically, in the second half of pregnancy, um, the bats with the low energy cost were able to spend less energy, right? Dealing with maybe the fungus. They had more energy for reproduction later on. In other words, the cost of tissue repair probably is as high for those bats that are in the higher growth colonies, and they had more energy to devote to reproduction, and thus they're a higher growth colony. That's what I believe is happening. It's kind of really important initial evidence on that connection. And it's interesting to note that the greatest cost appears to be during the time of tissue repair, not during the active response to fungus. So, it's pretty exciting, these results, and I'm working on publishing it right now. I'll just say one last point on this is the variation among colonies' immune response is not what we predicted. Now, just a brief little sense of the whole take-home message about where things are going with bats. So when you look at, oh, here you go. What happened? The bat population crashed as the disease came through. And then the question is, oh, geez, sorry about that. Are they going to hold on, right? As opposed to not going extinct? And how well will they do? Will some colonies grow more quickly or some colonies grow more slowly? And we know now that there's variation in how quickly the bat's recovering, as I've indicated. So then the question comes, what is the connection in the region between the hibernacula like Alice Cave and Eagle Cave, two important hibernacula in New York and Vermont, and there's also one down here, and these maternity colonies we've been studying, blue dots. We already confirmed the connections between them all. They definitely are connected. And that's part of research I'm doing now. This is demonstrating that we have been recapturing.
May I give you just a 5-minute warning? I'm so sorry,
Oh,
but I
okay.
don't want to miss the chance for people to ask you questions.
Yeah. I'm already taking a long time. It's all right. Thank you. All right. Thanks for that reminder. So this is about recovery. Recapture, and that there are bats that are recovery. And this is showing you how recovery rates are different. Some are recovering more quickly, again, than others. Now, just briefly, the main part of my research now is in the link between stress, the presence of coronaviruses, and immune response in wet nose syndrome. And these questions now are more broadly even looking at the population level. Like, what are the factors that are influencing how bats are recovering? And part of what's going on, and part of my study now, with the backing of the U.S. Fish and Wildlife Service, is to understand more about what might be good predictors of likely future growth, predicting which colonies are going to do better and which colonies are going to do worse. And what we did was we basically collected bat guano as an important way to look at these critical factors in a non-invasive way, like by taking guano. And then this has been used and is being used right now by me to model which maternity colonies in New England may be recovering more quickly versus which are not. Briefly, stress. Cortisol is an important measure. An estimate of stress is found in bat guano. We know that chronic stress could definitely reduce immune function and reduce, in particular, inflammation response. Cytokine response, very important measure. Cytokines are important immune proteins that communicate amongst their immune system components. And it's believed that, we'll see more pro-inflammatory cytokines in bats that are more actively dealing with white nose. And we want to use cytokine response as a predictor of how well bats might be responding to white nose at the colony level. Virus type and load. It's known that coronaviruses could be linked to increased susceptibility to white nose. And I played a role and continue to play a role in looking at things like astroviruses, which are important digestive virus, and building up a North American-wide bat library, a bat virus library, to get a better handle on what viruses are present and how they're impacting bats by looking at them in their bat guano. In the end, what I'm doing right now, is trying to look at how these different factors, cortisol levels, cytokine response, and virus presence or absence, is connected to future growth rates. And then using that to predict how well bats are going to do and can do out in the West, say in Washington State and other parts of North America. And that will help prioritize then how to protect more vulnerable colonies. If we think this colony of bat is more vulnerable, may not likely grow as quickly as another colony, we might want to spend more effort prioritizing that colony with like deploying vaccines, which are being developed right now, or probiotic treatments that can slow down the spread of the fungus, or enhancing the habitat for colonies that are more vulnerable by putting out insects in the habitat. That's being done right now. Just the last couple of things. There are a number of ways that the government's responding and the private industry as well. We're trying to contain the spread of the disease. We're trying to do ongoing research. We're trying to develop new management tools. We're trying to outreach to the community, the public, working with research managers, and of course the conservation community like yourselves about what can be done and what is being done to stop white nose. There's a national U.S. plan. There's a Canadian national plan. It's still being developed even now in this difficult time after Trump's been elected. We're still moving forward as best we can. And then I just have two more slides, I think, and I'll stop, Beth. Just want to show you there's a lot of levels of approach right now. We're trying to stop white nose and try to promote its recovery. And the last thing I want to note is that it's a very important concept. We're trying to study more what's happening in the bat, how it ties into PD. We're trying to understand how the environment affects the bat and its response to PD. And we're trying to understand more about how the environment affects PD and how PD affects the environment. And in disrupting this triad is an important way of trying to break down how PD is, affecting and killing bats. And then I think what I'll just do is go to there we go, acknowledgement slide. Just acknowledging some of the important people who've been very helpful in collaborating with me. I want to mention Tom did die tragically of, unfortunately, of COVID, but I also have a very wonderful memoriam that I wrote that I'll try to remember to send to you as well, Beth, that tells a lot about his life and passion for bats and might be worth reading. All right,
Yes,
thank you.
thank you. Oh my goodness. But
Mhm.
I have a really simple-minded question to ask you to start.
Sure.
And you showed all the photos of those bats, all the varieties of the species of bats that are here. When they fly overhead, can you tell what bat it is automatically?
Um, if it's close enough, that has happened when they pass pretty close, I can usually tell between a big brown and little brown. And that's only because they're one of the two common species you might see, flying. Um, I know that with the fruit bats, because their size is so different scale, people who I've talked to from like Australia or Papua New Guinea or Indonesia, what's happening in some bat researchers, you can tell pretty quickly what species is flying overhead. They fly more slowly. They have very distinctive color patterns, So it really depends on maybe where you are and what species are present. Yeah.
Okay, I live close to Crystal Lake here in Newton, and it used to be
Yeah.
you walked over there. My goodness. If you walked over there in the evening, you would see tons of bats flying overhead. And that isn't the case anymore. Um,
Hmm.
is that likely because of white nose syndrome or
I think so. I mean, it is true that there's a concern about insect levels in the planet. Parts of the world, insect levels, you may be aware of this,
That's
have
the very first question from the public was,
decreased independent of bats. We don't fully understand why. There's a number of, again, scientific hypotheses about this. Um, but I think white nose's impact is still pretty great. I mean, bats are coming back, and there's some parts of where I've gone back to where you see them again. But yeah, that's probably a lot of what it is. The numbers just aren't what they were since 2009.
okay. Another question we received is you mentioned something about people putting insects out for bats.
Yeah, it's a little bit, it's a fascinating idea. So one of the studies that's been done and been tried is the Bat Conservation International team working with U.S. Fish and Wildlife Service identified, I think, 3 locations in the Midwest and 3 in the Northeast where bats do what's called swarming. So one of their behavior patterns is the females and the males meet on the way between the maternity colonies and hibernacula, and they mate. At least the typical Vespertiliana bats, like little browns and big browns. And so when they swarm there, they often stay out for a few weeks. Um, they've been putting out just, yeah, literally putting out insects that they know they'll eat to give them a chance to fatten up even more, right, as they go into the hibernacula. That's right. That's what's been tried. Now, I know it's an ongoing study. It's not considered official protocol, but that is what's happened, yes.
Okay. And does that help? Another question that we have is how you assess how much is the reduction in insects and how much is the physiological symptoms of uh, results of the disease. Is there interest in trying to
Yeah.
psych that?
Yes. So I have done some preliminary insect sampling around the maternity colonies. I'd like to do more of it even now and look at is there differences in insect variability amongst these different colonies that are still at different growth rates. So there's a couple colonies of maternity colonies of bats. It's remarkable. They're up to 1,000 bats, when they were down to maybe 50 in 2010. And then there's some colonies that are just hanging on, 150, 190. And so I like to look at insect differences there and see if that is at all differences and just probably address that question. Yeah.
Okay. Now you showed us the hibernaculum that you went to,
Yep.
and it sounds like the ones you mentioned are a good distance away. Is that because that's where caves are and they're big caves? And here they're in barns or bell towers or
Yeah. Now, this is another big question mark, though. But first of all, yes, it seems like more of the classic, I think they're called karst caves, typical of New England. These are like things like quartz and other types of classic mixed rock minerals, are more central Mass, Western Mass, Southern Vermont, Eastern New York, Northern New York, up in the Cascade region, up White Mountains. Um, Probably that is why more of them are there. But we don't know. No one still really knows which caves are most important. How many maternity colonies are they going to 1 or 2 caves? Are they going to other caves we don't know about? Could it be there's also local movement, local bats living nearby? We just don't know. And I'm getting involved in a tracking study that's going to look at using cell phone technology. It's pretty cool. It's a new type of microchip about as big as my fingernail that you can put on a bat, and it sends out little signals that are picked up by anyone who wants to volunteer using their cell phone. And it's really easy to get on the network. And then that's it. You don't have to worry about it. And anytime a ping is picked up, it goes into this national database. And I'm going to probably start doing work on this if I get the money in the fall to look at more about where bats are going between hibernacula and
amazing. So how
yeah,
do you go into the hibernaculum and put
tag
the
'em.
sensor
Yep.
on them there?
Well, we might do both directions. Right now, the thinking is if I get some money, I'm working with some collaborators down in Martha's Vineyard.
Okay.
We capture some bats in the maternity colony probably in September when they're getting ready to leave. And then we try to track them. And the nice thing about it is they also have these little local, they call them nodes that you can plant near known hibernacula, which will also pick up their signal when they come by if you lose the cell phone signal. Yeah.
And how far do you think they will travel from Martha's Vineyard? Will they stay on the vineyard in that immediate area?
Well, some may stay and some may leave. That so we know that bats from Aeolus, as I showed you in that one picture, have gone as far down as the Cape Cod. They've gone down to Lincoln, Mass, where we know maternity colonies exist. They travel pretty far there. We also know that some of them seem to travel maybe locally, but again, that's part of what the study is. How many of them stay in Martha's Vineyard and locally hibernate if they can? How many don't? Big brown bats can definitely hibernate in um, wooden roosts or wooden spaces that are kept just above freezing.
Okay. Okay.
So they might be found on the edges of attics. People have found them in wooden pantries in their basement. They're cold. So big brown bats are very resourceful that way. Little brown bats typically have to go to hibernacula in the classic sense, either caves or mines.
Oh, fascinating. Well, you
Yeah,
will let us know if you decide you
yeah,
want us to have something on our cell phones, because that would be fun to do.
yeah. Well, we'll figure out. We're gonna develop an outreach plan,
Okay.
how to reach the whole public, and I'll definitely let you guys know. I'll let all the local conservation groups know. That's the idea. Yep. And there's a place you can sign up. It's gonna be easy. It'll be cool.
Terrific. Now we're going to go back a little bit and we're going to ask a very basic question. Can you say why bats are important? Why should the government support the reduction of white nose syndrome and PD?
So I think in a nutshell, with insectivore bats, they still are important pest reducers. I mean, in a nutshell, there's been now several studies have shown whether it's big brown bats or the Mexican free-tailed bat, which by the way has not been hurt that much by white nose syndrome because they're down near Texas and New Mexico, and white nose is only just getting there. But it's been demonstrated they eat a large number of pest species, pest beetle species, including invasive species, invasive moths. These are species that are known to be killing trees right now. A recent study I just read about confirmed that one of the number one ways deforestation is now happening in New England is from invasive species, even more than lumber activity, logging. So definitely important for reducing insect populations that are problematic. Um, Also, it's believed they are an important food source. Owls routinely eat bats. We didn't know that till more recently. So, that could impact owls and the role they play. Bats are at times eaten by those who are crafty, like weasels and fishers. That's right, they can get bats. Um, so they're also important prey. So in that sense, they're part of the whole food web. In that sense, they're important ecologically too.
Is it harmful if an owl eats a bat with the virus?
No, yeah, it doesn't seem. I mean, again, we're not 100% sure about this, but these viruses are very bat specific.
Okay.
Now,
So
I will say that I didn't mention this, but COVID, we're pretty confident, contrary to all the weird stories about what China did or did not do, came from a virus that came from a southeastern bat that was found in China called the horseshoe bat. And that bat, some of that virus jumped into a pangolin, and that virus mixed. That's right, into the with the pangolin virus and the horseshoe bat virus, and then jumped over to humans, probably in some wildlife markets. And that's how COVID became that coronavirus, um, SARS, whatever it's called, two, I forget what the exact full name of it. That official coronavirus is what probably caused COVID.
okay,
Yeah,
so there aren't any bat viruses that are in
no,
our area that will be transmitted to humans.
no. And when we wear those masks, and in many cases now we stopped doing it. I still do it in certain cases. It's because we're trying to stop the spread of PD,
Okay.
right? We're not worried about getting something from the bat. I've been in many, many caves before White Nose where we're wearing nothing, just walking around. Now, I would be careful to warn everyone. If you ever go into caves in the tropics, you can get histoplasmosis. That's a real threat, real problem, because they're bat fungus. I'm sorry, histo is a fungus, excuse me, that can grow in bat guano in the tropics. Anyway, quick
Okay,
note.
if we're lucky enough to get to go, we're going to
Yes,
check in with you.
yes.
Oh, Our President Catherine has a question for you that's very Catherine-like. Uh, should she leave her attic windows open to let bats get in?
Um, it's funny. I was talking to someone about this the other day because one of the hibernacula, I mean, sorry, one of the maternity colonies out here in Lincoln, the key entrance point is a window, the top loft of the barn that was cracked years ago. I don't forget when the owner took over, and he left it cracked forever when he realized the bats are coming and going. So that doesn't necessarily mean you should leave your windows open. Um, if it doesn't affect your heating bill and you have known bats that are roosting in your attic and you're okay with that, and there's some people and it can be contained in your attic and you keep a proper distance from them, then that may be a way to encourage their activity to go back and forth. But bats that start to get in attics, even the windows closed, find various cracks, openings. A lot of these older New England buildings are not perfectly sealed. They'll come and go anyway if they can do it.
Okay, interesting. We
Yeah.
have a question, what about rabies in bats?
Yep. That's always when I'm ready to say an answer about. So bats are number three, I think, of mammals in England that have the highest incidence of the incidence of the virus on average. I think it's raccoons and fox are higher, but bats definitely are carriers and can give you rabies, of course. In general, what I think is the advantage about knowing if a bat's rabid versus, say, a raccoon is if a bat is rabid and really seriously infectious, it's likely not to fly well and not very healthy and may not be easily able to get around. So what happens is if someone gets a bat in their house, the test to know what to do is typically I tell them leave it in the room, open the window, shut the door. And if the bat leaves, they'll definitely work very hard to leave. And they're gone within a day. They probably aren't rabid. They just got lost. If the bat's still flopping around, can't get around, could have rabies. Um, with that being said, you have to take the caution that they could have rabies. Um, but what's interesting about bats is they seem to have a higher incidence of rabies exposure and not dying.
Really?
Yeah. And no one knows why. So could they be more immune to rabies, which seems pretty remarkable? No one knows. Um, but there's another little factoid about that.
Thank you. I have more questions. We have more questions in our chat. Um, oh, I'll do one more and then we have to go.
Okay.
Um, are is the recovery different in different species? Are some recovering
Yes.
more than others? And
Yes.
did they develop immunity differently?
Yes. So just as a brief recap, I think I mentioned this, but I don't mind recap it. There's a slide where I present the 12 species that are first known to have white nose. Of those 12 species, I think three or four are doing far worse from responding to white nose than at least one or two. So, for example, big brown bats are probably the most successful at responding to white nose and dealing with white nose. Whereas the northern long-eared bat is probably the one of the most negatively impacted by white nose.
Okay.
So definitely, yep.
Okay. I've lied. I'm going to do one more. Have there been any behavioral changes noted in bat colonies to aid in the recovery or the avoidance?
Um, do you mean on their own?
Yeah.
I think that's hard to know. I think bats still seem to congregate in the same way, at least in the maternity colonies I go to, as they did before.
Okay.
They seem to still come together. The pups still are born around the same time. Oh, well, I'll take that back. But that's just they think it'll probably impact of climate change. That bats are born a little bit earlier because it gets warmer sooner.
Okay.
Um, but overall, I think at least the Northeast, no. Now, I will say bats in the Southwest, and I had a couple slides about that, but not a big deal, appear to be unevenly impacted by white nose. Some areas of New Mexico or Arizona have white nose and some do not. It's like very patchy. They think it's because some bats use caves different than other bats. So it could be differences in where they go that could be affecting which ones are getting white nose or not. If that makes any sense. So hibernation behavior, yes. Maternity colony behavior, no, I would say.
Okay. Thank you. Okay. We have questions, but we're going to stop now. Thank you. If there are people who have other questions for Dr. Richardson, you can send them to webinars@newtonconservators.org and we'll gather
Yeah,
them and we'll make sure they get to him. Thank you.
definitely.
And I will, as usual, send out information with all of the articles that Dr. Richardson has sent to us. You'll get it sometime tomorrow. We hope to see you again soon. And thank you so much, Chris. And good night, everyone.